My name is Joshua Mugabi. I welcome you to topic 8, growth in plants and development in insects. We are going to look on learning outcome A. Analyze the pre and post-germination stages during the growth and development of plants in relation to their significance in crop production. Learning activity. Part B. In groups, learners use microscopes to investigate cell size in the different regions of a young dicotyledonous root or shoot. They make their write-up and present it to the class. We are still going to look at investigating the cell size. Item 1. We want to look at the school garden observation. Students note that bean seedlings grow taller each day. Their biology teacher asks them to investigate which part of the shoot is responsible for the rapid growth using a microscope. Tasks. Identify the different regions of a young dicot shoot. Observe and compare cell size in each region. Record which region has the smallest and largest cells. Let's start with identify the different regions of a young dicot shoot. Regions include meristematic zone, elongation zone, and differentiation zone. Let's observe and compare cell size in each region. Cells in meristematic zone are small and densely packed. Elongation zone cells are longer. Differentiated cells are larger and specialized. Record which region has the smallest and largest cells. Smallest cells occur in the meristematic cell region. Largest cells are in the elongation and differentiation zones. We are still investigating the cell size. Item 2. We want to look at the farmer's curiosity. A farmer notes that roots of young bean plants extend quickly into the soil. He wonders where the cell division occurs in the root. Tasks. Prepare a temporary slide of a root tip. Observe cells near the root cap and further up the root. Compare the sizes of the cells. Let's prepare a temporary slide from the root tip. We we think that we have prepared, and we observe the cells near the root cap and those ones of far up the root, and we compare the sizes of those cells. Thin sections of the root tube are mounted and stained. Then the cells near the root tube are smaller and actively dividing, whereas the cells further away are larger due to elongation. We are still looking at investigating the cell size. Item 3, nail growth comparison. A student compares nail growth with plant growth and is asked to find the growth point in plants. Task: locate the meristematic region under the microscope. Measure cell size in the meristematic region. Explain why cells here are smaller. Let's locate the meristematic region under the microscope. Meristematic region is just behind the root cap. Then we can measure the cell size in the meristematic region. The cells are very small with large nuclei. We have to explain why cells here are small. Cells are small because they are actively dividing by mitosis. Still investigating the cell size. Item 4. We want to look at the kitchen experiment. A learner germinates beans in a jar and observes rapid root growth. Task: observe the root tip under low and high power. Identify regions with elongated cells. Explain how elongation causes growth. Let's observe the tip of the root under the low and high power of the microscope and identify the regions with elongated cells. Elongated cells are seen above the meristem. Elongation region has long, thin cells. Explain how elongation causes growth. Cell elongation increases the root length. Still under investigation of the cell size, item 5, we want now to look at the class practical test. Students are given an unknown slide of a young dicot shoot. The task is to identify the slide as a root or shoot. Label regions showing different cell sizes. Justify your identification. Let's start by identifying the slide as a root or a shoot. The slide is identified as a shoot by presence of a leaf primordia. Let's label the regions showing different cell sizes. Meristematic, elongation, and differential regions are labeled. Meristematic has the smallest, elongation region has largest and elongated, and differential region has those ones that are specialized and large. Justify your identification. Different cell sizes indicate different growth regions. We are still investigating the cell size, item 6, and we want to look at measuring cell size. Students are asked to quantify growth by measuring cells. Calibrate the eyepiece graticule. Measure average cell length in each region. Compare results. Let's start by calibrate the eyepiece graticule. Eyepiece graticule is calibrated using a stage micrometer. Measure average cell length in each region. Cells in elongation zones are longest. Compare results. Growth is mainly due to cell elongation after division. We are still investigating cell size, item 7. We want to look at a real-life transport analogy. Teacher compares plant growth to road construction. Task: identify where new roads or cells are formed. Identify where roads are extended. Match regions to functions. Identify where new roads or cells are formed. New cells are formed in the meristematic region. Identify where rods are extended. Cell lengthen in the elongation region. Match regions to functions. Differentiation region forms specialized tissues. Still under investigation of cell size, item 8, comparing root and shoot growth. Students compare root and shoot tip microscopically. Task: compare cell size in roots and shoots. Identify similarities and differences. Explain significance. Compare cell size in roots and shoots. Both have small meristematic cells. Identify similarities and differences. Shoots show leaf primordia. Roots show root cap. Explain significance. Both grow by cell division and elongation. Still under investigating cell size, item 9, daily observation journal. A student keeps a growth diary of a bean seedling. Task: relate daily height increase to microscopic observation. Explain which region contribute mostly to growth. Relate daily height increase to microscopic observation. Increase the height corresponds to cell elongation. Explain which region contributes most to growth. Elongation region contributes most to rapid growth. We are still under investigation of cell size. Item 10. We are looking at practical examination setting. During exams, students are asked to interpret microscope observations. Tasks: sketch cells from different regions. Compare cell size in your drawings. Draw conclusion. Sketch cells from different regions. Meristematic cell drawn small and compact. Compare cell size in your drawings. Elongated cells drawn long and thin. Draw conclusion. Growth occurs by division and elongation of cells. Thank you very much for participating in Topic 8, Growth in Plants and Development in Insects. We are on Learning Outcome A. Analyze the pre- and post-germination stages during the growth and development of plants in relation to their significance in crop production. We have been looking at learning activity B. In groups, learners use microscopes to investigate cell size in the different region of a young dicotyledonous root or shoot. They make a write-up and present to the class. If this has been very useful, visit me on Mugabi Joshua Biology on YouTube.